Hello, I'm Professor Len Adelman of the University of Southern California. I thank the organizers of this conference for providing me this opportunity to address this distinguished group. The title of my talk is The Tiniest Treasure Chest, and what I want to describe is what I think is a revolution in science and engineering, which is just now emerging, a revolution that will change many aspects of our future. The key to this revolution is the tiniest treasure chest, and here is a picture. The tiniest treasure chest is the cell, and the treasures inside it are the molecules you find there. So not the simple molecules like water or salt, but rather highly complex molecules which have arisen over billions of years to do very special jobs and do them extraordinarily well. I have a short video which shows some of those molecules in action. It actually shows the process of DNA replication. This is an important process inside the cell, since it's the replication of DNA that ultimately allows cells to replicate, and it's the replication of cells that ultimately allows you and me to replicate. It's not important to remember the details of this animation. Rather, it's just important to get a sense of what these molecules look like and what they do, and also to see that it might be reasonable to think of these molecules as tools or small machines. So let's see the animation.
When DNA replicates, its strands are separated by the enzyme helicase. Single-stranded DNA binding proteins keep the strands from re-annealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase 3. No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then, DNA polymerase 3 lays down new DNA. The process repeats again and again. DNA polymerase 1 replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragments.
I want to now describe some of the properties that these molecules have which are so extraordinary. Here's a picture of the polymerase molecule, the one we saw copying DNA in the animation. It's extremely small, as are all the molecules. It's a few nanometers in each dimension. A nanometer is 1/1000th of a meter. But to get a better sense of how small that is, here's a tube that contains 1 milliliter of fluid. Now, if I were to fill the same volume with polymerase molecules, I would have an extraordinary number of them because they're so small. In fact, I'd have approximately 5 times 10 to the 18th such molecules in that volume. That's 1,000,000 times 1,000,000 times 1,000,000 times 5. A remarkable number of molecules in such a small space. In addition, these molecules are extremely energy efficient. Here is ligase, the molecule you may remember in the animation that takes 2 strands of DNA and sticks them together. Ligase, like all the molecules inside the cell, has evolved in an environment where it was very important to use energy efficiently. If one cell uses energy twice as efficiently as another, then that cell has to only find half as much food to stay alive. And this has pushed the energy efficiency of cells through the billions of years to the utter brink of what thermodynamics says is possible. So here is a weight. This is a 1 kilo weight, and if I were to raise it 1 meter up, I've used 1 joule of energy. Now, how many operations could ligase do with a joule of energy? Where an operation means sticking 2 pieces of DNA together permanently. The answer is, with 1 joule of energy, ligase could do an astonishing 20 times 10 to the 18th operations. That's 1,000,000 times 1,000,000 times 1,000,000 times 20. Incredible energy efficiency. These molecules, some of them store information, and they do that extraordinarily well. They have extreme information density. So here is the famous double helix, DNA. It stores information. In fact, it's been storing the so-called blueprint of life for billions of years. But how much information does it store? Here is actually a single gram of DNA. This is 1 gram of DNA. It's about 1 cubic centimeter. I'm going to show it on a close-up here so that you'll have a better sense of what it looks like. Now, there's a lot of information that can be stored in 1 gram of DNA. We tend to think that CDs are efficient ways of storing information. But which can store more, 1 gram of DNA or this CD? Well, it turns out that it's really no contest. The 1 gram of DNA can store approximately as much information as on 1 trillion CDs. So in terms of bits, the amount of information that can be stored in that 1 gram of DNA is 4 times 10 to the 21st approximately bits. That's 1,000,000 times 1,000,000 times 1,000,000 times 4,000 bits. This is so much information that can be stored that if we do any reasonable estimation of all the information generated by mankind since the start of time, it could all be stored very easily in that 1 gram of DNA. So here it is, the tiniest treasure chest, full of miraculous molecules. Molecules that do amazing things. Molecules that are actually used to build you and me. We're not just made of these molecules, we're created by these molecules. And this is an untapped legacy of 3 billion years of evolution. These molecules are so extraordinary in their properties and in their function that they're way beyond our ability to create similar little machines de novo. But through the efforts of molecular biologists over the last 50 years, we've reached a point where we can take these molecules out of the cell and make them commercially available. So this confronts us with a challenge. We now have these wondrous molecules, these tools. What can we build with them that will enhance our future? And groups of scientists around the world are beginning to explore that possibility. What can we build with them? Could we, for example, build a computer? Well, that seems unlikely at first glance, since computers are made of metal and plastic and are basically non-organic, are sort of anti-biological. And the molecules we're talking about are biological by their very nature. But mathematics done in the 1930s may give us hope that perhaps we can compute with these molecules. At that time, the great logicians developed a theory of computation, which, interestingly enough, preceded the advent of actual electronic computers by about 10 years. And they taught us much about the nature of computation. In particular, they taught us that computation is easy. That to do computation, you just need a method for storing information and a few simple operations for acting on that information. We've grown accustomed to thinking that the method of storing information should be electronic in terms of binary, in the form of a memory RAM or in the form of a CD or a hard drive. And we've grown accustomed to thinking that the memory is acted on by operations we find inside of a processor chip, in this case, an Intel Pentium chip. And the remarkable thing is that with these two tools, we can do amazing things. If we start with the right information in the memory and we act on it with the right sequence of operations, that is, write a program, we can do things like play chess as well as anybody in the world can play chess. And we can guide a rocket to Mars, or we can balance a checkbook. In fact, the mathematicians have proved that these two components are so good at computing that they're so-called universal. They can compute anything that's computable at all. But we've seen that the molecules inside the cell can store information and do it extraordinarily well. And we've seen that some of the other molecules, like polymerase or ligase, act on that information. Polymerase will take a molecule containing information and make a copy that contains the same information. Ligase will take two molecules with information and stick them together into just one strand of information. Is this enough to compute? Well, mathematically, we can actually show it is enough to compute. And not only is it enough to compute, it's universal as well. Anything that's computable at all can be computed with these tools, in theory. But what about in practice? Can we actually do a computation with the molecules inside the cell? Well, in 1994, it was shown that the answer is yes. And so in 1994, a computation took place using the molecules inside the cell. The actual computation took place in this kind of tube. The tube's about 2 centimeters high, and it took place in a few drops of water at the bottom of that tube. It's called the DNA computer. It's much different than the computers that we're accustomed to, the electronic computers. They're solid and based on electronic principles. This is actually a liquid computer. The computation takes place in solution, and it's based on chemical processes. Now, I want to give you some sense of the problem that was solved on the DNA computer in 1994. It's a problem called the Hamiltonian Path problem. But I want you to see it because I want to show that this is not a problem having anything to do with biology. This is a problem having to do with mathematics and computation. So consider the following diagram. Think of the 7 balls shown here as cities, and think of the arrows going between them as indicating airline flights between the cities. And you would like to take a trip. And your trip, you want to begin at the green city and end at the red city. But during the trip, you'd like to visit each of the 5 blue cities. Is there a sequence of airline passages which will allow you to accomplish this? A so-called path. And the answer is yes. Here's a path that will do it. We could start our first flight there, then go there, then there, then there, 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 and there. So we've started at the original city, ended at the desired end city, and visited all the 5 blue cities as well. But we might not feel perfectly good about this path in that we might object to the fact that this city we visited twice. So one of the blue cities we actually had to return to. And maybe we didn't want to do that. So we might look for a better sequence of airline flights. Is there one that will start at the green city, end at the red city, and visit each of the cities without ever returning to one? And actually the answer is, in this case, yes. Here's the original graph, and that sequence of flights will take you to each of the cities exactly once, starting at the green city and ending at the red. Such a perfect flight plan is called a Hamiltonian Path. And so this is the problem that was solved in that 1994 experiment. Given this diagram, find the Hamiltonian Path. And it was found. So how did it work? Well, remember that DNA stores information. So we can take the information in this diagram, the things you see in this diagram, and we can store it in some strands of DNA. And then that DNA is added to the tube. We then act with various molecules taken from the cell on the DNA in the tube and apply various procedures to the DNA in the tube in such a way that we destroy some of the DNA and we create new DNA. And after the entire process is done, if we grab the DNA that's left, it will turn out that it's DNA which encodes the information in our desired Hamiltonian path. And that's how the process proceeds. How good was this 1994 computer? Well, in many ways it was very good. It could be argued that in 1994 this was the fastest computer in the world. It could also be argued it was the most energy efficient computer in the world. Now, scientists are working now to try to scale up this small computer to solve real problems and perhaps to compete with electronic computers. For that to happen, a lot of technical obstructions are going to have to be overcome, and whether it'll happen or not is a research question still to be answered. But the point is it did compute. So we'll end where we began, with the tiniest treasure chest, this legacy of evolution, this collection of wondrous tools. We know that these tools can build human beings. We've seen that even though, as scientists, we're very clumsy with the use of these tools right now, we can still build a computer. So it's reasonable to expect that as we get better with these tools, we'll be able to build new and exciting things. Things that will change the way we do medicine, change material science, change the way we manufacture things, change virtually every aspect of our future. Thank you.